Good morning, students. Today's class topic is protein targeting, sorting, and its associated disorder. In today's class, you are going to learn about what is meant by protein targeting and sorting, and what are the protein targeting disorders, and what is meant by prion and prion disease. Let's consider we have four scenarios. In scenario number one, let's assume this is a beta cell. So from beta cell, from rough endoplasmic reticulum, insulin will be produced. Once insulin is produced, it will be stored and it will be secreted out of the cell. So in scenario number one, once a protein is produced, that protein has to be secreted out. Let's consider scenario number two, and let's assume now it's a skeletal muscle. So in this cell, once an insulin receptor, which is a protein, once it is produced, it has to be integrated in the cell membrane. Then only the insulin can act on this particular cell. So in scenario number two, once a protein is produced, this protein has to be integrated in the cell membrane. Let's assume in the same cell, for glycolysis, we are going to produce enzyme from rough endoplasmic reticulum. So enzymes are protein. So once a protein is produced, that is the enzyme which is involved in glycolysis is produced, it has to stay in the cytoplasm because its work, that is its function, is in the cytoplasm. The metabolism, the glycolysis is going to take place in cytoplasm. So in scenario number three, once a protein is produced, that protein has to stay in the cytoplasm. Let's take another scenario where a protein which is required by lysosome is produced, so it has to be transported to the lysosome. So we had discussed four scenarios. So in the first scenario, the protein has to be secreted out. In the second scenario, the protein has to become a part of the cell membrane. In the third scenario, it has to stay in the cytoplasm. That is, the protein has to stay in the cytoplasm. In the fourth scenario, the protein has to be transported to the lysosome. So once a protein is produced, the cell has to decide whether that protein has to be transported out, or it has to become a part of the cell membrane, or it has to stay within the cytoplasm, or it has to become part of the, that is, it has to be transported to different cell organelle. This process, this type of sorting is called as protein sorting and targeting and sorting. So the cell, how the cell decide which protein has to be transported out or to different part of the cell is called as protein targeting or sorting. So there are two types of protein targeting and sorting. So when a protein is produced, that is during translation itself, if the protein decide this protein has to be transported to this particular organelle, then it is called as co-translational translocation, otherwise called as rough endoplasmic pathway. If once the protein is produced, only after protein is produced, if the cell decide this protein has to be targeted to, that is sorted to this particular organelle, then it is called as post-translational translocation or cytosolic pathway. So we have two different types of protein sorting, that is co-translational translocation and post-translational translocation. First, we'll see about co-translational translocation. So let's assume this is a cell and this is the nuclei of the cell and this is the rough endoplasmic reticulum. And we have lot of ribosome which is present in cytoplasm, that is free ribosomes. So let's concentrate on only one ribosome and rough endoplasmic reticulum. So in this scenario, that is in co-translational translocation, the translation will start in free ribosome. So we have free ribosome. Free ribosome means it is not attached to the rough endoplasmic reticulum. We have free ribosome. So the free ribosome will start producing polypeptide chain. That is, it is going to be involved in translation. So now it is going to start producing polypeptide chain. The initially produced polypeptide chain is called a signal peptide. Then the actual polypeptide chain will start producing. That actual polypeptide chain means, so from here, that is that from here green component only, the actual polypeptide chain is produced, and that is going to be folded to form the protein of our interest. So the initial polypeptide chain, the initial few amino acid is called a signal peptide. So why it is important? Once the signal peptide is produced, to this signal peptide, a signal recognition particle, SRP, signal recognition particle, will get attached to it. So what is the function of this signal recognition particle? This SRP is going to drag this complex, that is the ribosome, along with the growing polypeptide chain towards the receptor which is present in the rough endoplasmic reticulum. So this SRP, once it get attached to the signal peptide, it is going to drag this complex towards the rough endoplasmic reticulum, and it is going to attach the ribosome to the rough endoplasmic reticulum. So when this process occurs, this growing polypeptide chain is going to enter the rough endoplasmic reticulum through the pores which is present in the rough endoplasmic reticulum. Once this process has occurred, this SRP will get separated and it will start searching for a new signal peptide, and then it will drag that particular ribosome along with the growing polypeptide chain to the rough endoplasmic reticulum. So it will keep on doing the same process again and again. So now the ribosome is attached to the rough endoplasmic reticulum, and this polypeptide chain will keep on growing. That is, the translation will continue and the entire polypeptide chain will enter the rough endoplasmic reticulum. Now the entire polypeptide chain has entered the rough endoplasmic reticulum. The purpose for this signal peptide is over. So we are going to remove this signal peptide. So we are going to remove it using an enzyme. The enzyme is going to break the signal peptide. So what will be the name of the enzyme? It is going to remove the signal peptide. So it is called a signal peptidase. So signal peptidase will remove the signal peptide part of the polypeptide chain. Now we are having only the polypeptide chain of our interest. So this polypeptide chain will fold to form the functional protein. But the problem is, similar to this ribosome, we have multiple ribosome which is going to get attached to the rough endoplasmic reticulum, and from this ribosome the polypeptide chain and one more polypeptide chain will be produced. So there is a possibility for this two polypeptide chain to form bond to form an abnormal folded protein. So we don't want that to happen. So what we are going to do? We are going to cover this polypeptide chain with a protein called as chaptron. So what is the function of chaptron? Chaptron will prevent the interaction of the polypeptide chain with the nearby polypeptide chain. So it is providing an environment in which the polypeptide chain can fold properly to form a functional protein. So remember, chaptron does not cause folding. It is going to provide an environment in which the polypeptide chain will fold to form a functional protein. So now the chaptron has prevented this interaction. This polypeptide chain, now it can able to fold to form a functional polypeptide chain. So not only folding will occur, several other modification will occur, result in formation of functional polypeptide chain. So what are the other modification which is going to occur? So the other modifications are, apart from protein folding, we also have assembly into multiple subunit complex, disulfide bond formation will be there, glycosylation, addition of glycolipid, so that this protein will be targeted towards plasma membrane. So these are the other modification which will occur in rough endoplasmic reticulum. Now the polypeptide chain has folded and it has undergone modification to form a functional protein. So now the cell will decide whether this protein has to remain within the lumen of the endoplasmic reticulum, point number 1, or else it has to be integrated into the membrane of the endoplasmic reticulum, point number 2, or else it has to be transported to Golgi apparatus. From Golgi apparatus, whether it has to be sorted to cell membrane or outside the cell, or it has to be transported to different organelle. So how the cell decides where it has to be transported, that is where this particular protein has to be transported. So it does this by pasting a label over the protein. So it is going to paste a label over the protein. You can imagine this like you are writing an address over an envelope. So when you write an address over an envelope, based on the address, that particular envelope will be transported to that particular place. Like this, the protein will decide this particular protein has to be transported where. Based on that, it is going to paste a label over the protein. Based on the label, it will be transported. That is, it will be sorted. So what are those labels? So the labels are nothing but the targeting sequence. So if the cell decide that particular protein has to remain within the endoplasmic reticulum lumen, then it will attach a signal peptide sequence in the N-terminal end. If the cell decides the if the cell decide if the particular protein has to be integrated into the membrane of the endoplasmic reticulum, then it will add an KDEL sequence. to the amino terminal end of that particular protein. And if the cell decide it has to be transported, that is that particular protein has to be transported to Golgi membrane, then it will add diacidic sequence. If the cell decide if that particular protein has to be transported to mitochondria, then it will add a 20 to 80 particular sequence of amino acid at amino terminal end. And if that particular protein has to be transported to nuclei, the protein we will add nuclear localization signal. If that particular protein has to be transported to peroxisome, we are going to add peroxisome targeting signal. And if we want to transport it to lysosome, mannose-6 phosphate will be attached to the protein. So by this method, by adding different label like this, the cell will do the sorting and targeting function. That's all about co-translational translocation. Most of the proteins are sorted by co-translational translocation. Only few proteins are sorted by post-translational translocation. So next we are going to see about this cytosolic pathway, otherwise called as post-translational translocation. In this pathway, that is in this method, post-translational translocation, first the protein is completely produced by the free ribosome. That is, the ribosome will produce the entire polypeptide chain. Then it will undergo folding to form a functional protein. Once the functional protein is produced, the cell will decide whether this functional protein has to be targeted, that is sorted to nuclei or to mitochondria or to lysosome. So only to this three organelle it can be sorted by this method. So by this method, we can able to sort protein either to nuclei or mitochondria or peroxisome. So which marker, that is label, we are going to use the same label which we had mentioned in co-translational translocation. We are going to write here also. That is, if the cell decide if that particular protein has to be transported to nuclei, then it will attach a nuclear localization signal. And if that protein has to be transported to mitochondria, we are going to attach a 20 to 80 amino acid, which is rich in serine, threonine and basic amino acid. And if that particular protein has to be transported to peroxisome, we are going to attach peroxisomal targeting signal. So by this method, that is by post-translational translocation, we can able to translate it, that is sort the protein only to this three organelles. So that's all about co-translational translocation and post-translational translocation. So we had seen about protein targeting and sorting. Next, coming to protein targeting disorders. There are 7 important protein targeting disorders which we had read in different metabolism. We are going to revise it in this topic. So those disorders are eye cell disease, Zwellinger syndrome, infantile refsum disease, hyperoxaluria, cystinuria, familial hypercholesterolemia, and cystic fibrosis. We'll see one by one. Starting with eye cell disease. So in eye cell disease, the defect is in the enzyme N-acetyl glycosamine phosphotransferase. So this enzyme is required for the conversion of mannose to mannose-6 phosphate. So now only we have learned in protein sorting that mannose-6 phosphate is required for the transport of protein to lysosome. So now this mannose-6 phosphate is not produced. So the enzyme which is involved in the digestion of oligosaccharide, lipids and proteoglycan in lysosome is not transported to lysosome. So there will be accumulation of oligosaccharide, lipid and phospholipid. Since mannose-6 phosphate is not produced, the enzyme which is involved in breaking down this molecule is not transported to lysosome. So this molecule will be accumulated in lysosome. Next, Zwellinger syndrome. So let's see what happens normally in the cell. So in normal cell, the free ribosome will start producing enzyme which is required for the catabolism of long chain fatty acid and branch chain fatty acid. Once this enzyme is produced, to this enzyme a peroxisome targeting signal will get attached. To this complex a protein will get attached, that is peroxin protein will get attached. So this peroxin protein will guide this enzyme towards the peroxisome. Once the enzyme has entered a peroxisome, within the peroxisome, the enzyme will be involved in the catabolism of long chain fatty acid and branch chain fatty acid catabolism. So this is what happens normally. But in case of Zwellinger syndrome, there is a mutation because of which peroxins are not produced. So since peroxins are not produced, the enzymes which is involved in very long chain fatty acid and branch chain fatty acid catabolism, which has to be transported to peroxisome, will not be transported. That is, it will not be sorted. So that will result in accumulation of long chain fatty acid and branch chain fatty acid within peroxisome. So in Zwellinger syndrome, there is a mutation which result in reduced or defective production of peroxin, which result in accumulation of long chain fatty acid and branch chain fatty acid within peroxisome. Next, infantile Refsum's disease. So this infantile Refsum's disease is an less severe form of Zwellinger syndrome. So same thing, that is, there will be a mutation of gene which result in reduced production of peroxin, which result in accumulation of long chain fatty acid and branch chain fatty acid. So till now we had seen about eye cell disease, Zwellinger syndrome and infantile Refsum's disease in protein targeting defect. Next, coming to hyperoxaluria. We have learnt about hyperoxaluria in glycine metabolism disorder, right? So there are two types of hyperoxaluria, primary and secondary. This protein targeted defect related to hyperoxaluria is due to primary hyperoxaluria, which is an autosomal recessive disorder where there is a defect in the transport of alanine glyoxylate transaminase. That is, this enzyme normally it has to be transported to peroxisome. Instead of that, it is transferred to mitochondria. So in peroxisome, we don't have this enzyme, alanine glyoxylate transaminase. This enzyme is required for the transamination reaction between the glyoxylate and alanine, result in formation of glycine and pyruvate. Since we have a defect, that is, the enzyme is not transported to peroxisome, the glyoxylate will not get converted to form glycine. So glyoxylate will get accumulated and it will enter an alternate pathway. So this excess glyoxylate will get reduced to form glycolate and oxalic acid. So oxalic acid concentration in the blood will increase, that will get excreted in the urine, and that's why it is called as hyperoxaluria, because in urine the oxalic acid is excreted in excess, because of which the oxalic acid will form crystal, result in renal calculus and hematuria. So the treatment will be increased water intake and reduce food consumption which contain oxalic acid in it. That is excess amount of oxalic acid in it. So that's all about hyperoxaluria. So we had seen about eye cell disease, Zwellinger syndrome, infantile Refsum disease and hyperoxaluria. Next, coming to cystinuria. So we had read about cystinuria in sulfur containing amino acid metabolism disorders, right? So cystinuria is one of the Gerard tetrad. It was discovered by Gerard, and in cystinuria there is a defect in the absorption of cysteine, ornithine, lysine and arginine. So how to remember? CoLA. Okay, so all this amino acid in intestine as well as reabsorption of this amino acid in renal tubules. So when cysteine is getting excreted in the urine in excess, two cysteine will combine by disulfide bond to form cysteine. Cysteine is water soluble, cysteine is water insoluble. So it will combine to form crystal, which result in a renal stone formation. So the investigation, nitropuricate test will be positive. We have to hydrate the patient and alkalinize the urine to prevent cysteine formation. So that's all about cystinuria. Next, coming to familial hypercholesterolemia. Here the defect is in transport of LDL receptor to the cell membrane, that is the surface of the cell in liver and other tissue. So there will be no LDL receptor present in the surface of the cell. So LDL will not be taken up by the cell. So LDL will keep on circulating in the blood. So the LDL will not be cleared. So the LDL concentration in the blood will be increased. So the main function of LDL is to transport cholesterol. So cholesterol is not cleared. So cholesterol concentration in the blood will be increased. That will result in increased risk of atherosclerosis. Finally, coming to cystic fibrosis. The problem in cystic fibrosis is there is a mutation in CFTR gene, that is cystic fibrosis transmembrane conductance regulator gene. So this gene is required for the production of chloride channel. So now this chloride channel is not produced in the epithelial cells. So when there is a defective chloride channel, chloride is not transported. So water is not transported out. So the mucus will become sticky. That is, it is less in water, so it will become sticky in lung, pancreas, so that this will cause a repeated infection. So the patient will present with clinical features of repeated respiratory tract infection. The patient will be having chronic productive cough, failure to thrive, pancreatic insufficiency, and male infertility. The investigation will be, the gold standard investigation is to do sweat chloride test, in which chloride concentration will be very high. So that's all about protein targeting and sorting and protein targeting disorder. Next, coming to prion and prion disease. PRP protein is a normal protein which is present in leukocytes and nerve cells. It is a sialoglycoprotein which is anchored to the cell surface. So this protein, PRP protein, will undergo structural modification to become abnormal prion protein, PRPSC protein. So this abnormal protein is resistant to heat and proteolytic enzyme. So if you see, this is the normal PRP protein, this is the abnormal prion protein. So if you see the structure, the normal protein is made up of only alpha helix, the abnormal protein is made up of alpha helix and beta pleated sheets. So this is the structural difference. But if you see the amino acid sequence, it will be exactly same to that of the normal. That is, the abnormal prion protein will be having the same amino acid sequence to that of the normal one. So what is abnormal prion protein? That is PRPSC. Compared to the normal PRP, this abnormal PRPSC will be having similar primary structure, but they will be different in their secondary and tertiary structure. That is compared to the PRP protein. So how a patient will develop prion? Either the gene should be mutated, that is the gene which is involved in PRP production will be mutated, or else a person, if the person get injected or ingested the abnormal protein, this abnormal protein will convert the normal PRP to abnormal PRP protein. That is normal PRP to abnormal prion protein. So once the abnormal prion protein is formed, this abnormal prion protein will convert other nearby PRP protein to abnormal PRP protein, PRPSSAC protein. So they are infectious in nature. This abnormal prion protein cannot be digested by lysosomal enzymes. So they will accumulate within the cell, result in cell death and neuronal degeneration. So because of neuronal degeneration, the patient will have developed dementia, neurological defect, ataxia. And this is a slowly progressive disease, but later it will become fatal. Prion disease can infect both human and animal. So these are some of the disease which is seen in animals. That is prion disease which is seen in animals. That is crepey, bovine spongiform encephalopathy and chronic wasting disease. Some of the examples of prion disease in humans are Creutzfeldt-Jacobs disease, fatal familial insomnia and Kuru. So to summarize, we have seen about how proteins are sorted by endoplasmic reticulum pathway and cytosolic pathway and disorders related to it, and prion disease. Thank you.